I'm pleased to witness such a varied and involved group of participants. Your presence contributes greatly to the depth our discussions and highlights our shared dedication to progressing in our respective areas of expertise. I look forward for fruitful interactions and mutual learning as we embark on this educational journey. SPSR is acclaimed for its engaging webinars, further establishing as a leading platform for continuous professional growth within the pharmaceutical field. I am certain that today's webinar will uphold our tradition of excellence, presenting valuable perspectives from a distinguished presenter. Let's embrace this chance to gain a fresh knowledge, participate actively, and move our industry forward. Welcome everyone. I look forward for beneficial experience for all participants. We are gathered today to commemorate the 60th SPSR webinar on animal models and neuropsychiatric disorder research, Building the Bridge, a distinguished event in the series hosted by Society of Pharmaceutical Sciences and Research under the esteemed guidance of Dr. Shashi Alok sir, National President SPSR. The Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is committed to the progression and enhancement of pharmaceutical field through diverse strategies. We aim to facilitate conferences that promote pharmacy-related initiatives and endorse programs and activities addressing public health issues in India. Central to our purpose is the commitment to fostering the growth and rich exchange of information regarding pharmaceutical practices and services. By pursuing these goals, we strive to raise both the quality and influence of pharmacy profession, cultivating a proactive and cooperative community. It's a great privilege to introduce Dr. Kunjbihari Sulakhiya sir, Assistant Professor Pharmacology, Neuropharmacology Research Lab, Department of Pharmacy, Indira Gandhi National Tribal University, MP, India, as a distinguished speaker today. Dr. Kunjbihari Sulakhiya sir's expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussions and expand exploration of neuropsychiatric disorder research. We are grateful for his engagement and anticipate an enlightening sessions. It's a privilege to introduce Mrs. Monika Sabharwal, ma'am, as a convener of this webinar, who also serves as the National Secretary of SPSR and the Managing Editor of both IJPSR and IJP. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are grateful to have her guidance in today's session. I'm pleased to invite Mrs. Monika Sabharwal, ma'am, to warmly welcome our distinguished guest speaker. Monica, ma'am, over to you. Monica, ma'am, am I audible?
I am Monika Sabharwal, the convener of this pleasure to welcome you to today's session titled Animal Models and Neuropsychiatric Disorders Research Building the Bridges. This webinar, organized by Society of Pharmaceutical Sciences and Research SPSR, marks a significant milestone in our ongoing journey to advance the knowledge in the research in the field of pharmaceutical sciences. We are honored to have Dr. Kunjbihari Sulekhiya sir as our esteemed speaker for this event. Dr. Sulekhiya served as an assistant professor of pharmacology at Neuro Pharmacology Research Lab (NPRL) within the Department of Pharmacy at Indira Gandhi National Tribal University (IGNTU), Amarkantak, MP, India. His extensive experience and the research in the neuropharmacology have made him recognized authority in the field. particularly in use of animal models to study neuropsychiatric disorders. Today's topic is both timely and very critical. The use of animal models in a neuropsychiatric disorder research is a cornerstone of understanding the complex brain functions and the underlying mechanism of mental health conditions. Dr. Sulakhiya will guide us through the intricacies of how these models bridge the gap between the basic research and their clinical applications, offering insights that are crucial for developing the effective treatments and the therapeutic strategies. With over 2,000 registrations from more than 15 countries, this webinar has drawn a truly global audience, reflecting the universal importance of neuropsychiatric research. The overwhelming interest and the participation in our webinars underscore the value of these discussions in shaping the future of healthcare and pharmaceutical sciences. As we celebrate the 60th SPSR webinar, I would like to extend my heartfelt thanks to our global community. Your continued support, whether through the active participation, insightful feedback, or the regular attendance, have been driving force behind our success. It is this collective effort that empowers us to strive for the excellence and bring forward the best pharmaceutical education and the research. Before we begin, I would like to extend a very warm welcome to Dr. Shashi Alu, our National President, whose visionary leadership has been pivotal in the remarkable transformation within the SPSR. I will also like to acknowledge the tremendous efforts of our entire SPSR team, organizing such wonderful events on regular basis, particularly on the weekends, is no small feat and dedication. The hard work of everyone involved do not go unnoticed. Your commitment is what makes these webinars a source of knowledge and inspiration for all our audience. Our goal with this webinar is to provide a platform for knowledge sharing and professional growth and response we have received that motivate us to keep pushing forward. We are committed to make each webinar better than the last one, ensuring that we deliver a valuable content that make this positive impact on our profession. I would like to extend my heartfelt thanks for the significant contributions made by Mr. Junaid Tantray, the co-convener of this webinar, alongside the organizing committee members and the all SPSR team. Their relentless dedication has been instrumental in bringing the vision of this event into fruition. The seamless execution of this webinar is a testament to outstanding effort of our team working behind the scenes. Their dedication and high quality work and persistent effort have been pivotal to a success of every event we organize. My gratitude goes to each member of SPSR team for their careful management of the details and enduring enthusiasm that brought this webinar a future. Stay ahead in the field of innovation with the SPSR. Join our YouTube channel for unique webinars, experts' perspectives, and the recent findings in the field of pharmaceutical sciences. Engage with our vibrant community that is involved in the conversation driving the healthcare progress for all of us. Keep to update by subscribing the YouTube channel and turning on the notification bell icon for immediate alerts. Follow our progress in advancing the pharmaceutical sciences that will benefit the society. As we begin this session, I encourage everyone to engage actively and absorb the wealth of information that the Dr. Sulakhiya will share. Let us use this opportunity to build the bridges in our understanding of the neuropsychiatric disorders and explore the new avenues for research and collaboration. Thank you for joining us today, for your continued support of SPSR's initiative. Over to you, Mr. Junaid Tantray.
Thank you, Mrs. Monika Sabharwal, ma'am, for granting me this opportunity. Now we'll start with a short introduction to guest profile. Dr. Kunjbihari Sulakhiya, sir, is an esteemed assistant professor in the Department of Pharmacy at Indira Gandhi National Tribal University, Amarkantak, MP, India. With the robust academic background, he obtained his MS and PhD in pharmacology and toxicology from National Institute of Pharmaceutical Education and Research, Naipur, Guwahati, Assam, India. Dr. Sulekhiya sir has made significant contributions to the field of neuropharmacology through his extensive research focused on exploring plant-derived bioactive molecules for the treatment of anxiety, depression, autism, and cognitive dysfunction. His dedication to advancing knowledge in this area is evident from his impressive publication record, which includes 17 research papers, 7 review articles, and 10 book chapters in a reputed peer-reviewed international and national journals. Dr. Sulekhiya sir has been recognized for his advanced, outstanding research through 12 awards and fellowships, including 6 travel grants, allowing him to present his research at prestigious international conferences in the USA, Canada, Japan, France, and Macau. His expertise and contributions have earned him membership in several prominent professional societies, such as Indian Academy of Neuroscience, International Society for Neurochemistry, and the Asia Pacific Society for Neurochemistry and APTI. In addition to his research and teaching roles, Dr. Sulekha sir serves as an editorial member and reviewer for various reputed journals, further showcasing his commitment to the advancement of science and his field. It's a great satisfaction and pride that we declare that we have exceeded 2,000 registrants globally, showcasing an impressive diversity of countries. Significant attendees is noted from the nations like India, Bangladesh, Cambodia, Ghana, Indonesia, Kenya, Kuwait, Malaysia, Nigeria, Saudi Arabia, Sudan, and all other countries. We are deeply moved by the substantial and very response we have gotten. Thank you all for joining us today. Let's not waste more time. Let's make this webinar insightful and enriching experience for everyone involved. We have esteemed keynote speaker, Dr. Kunjbihari Sulekhiya sir with us. Welcome, sir. Please share your presentation, sir.
Thank
Yes, sir.
you,
It's okay.
Dr. Junaid. Is that okay?
Yes, sir. It's totally okay, sir.
Okay.
Please continue.
So, very good morning to all of you. I'm very thankful to the President of SPSR, Dr. Sasialo, National Secretary, Dr. Monika Sabharwal, Madam, and co-convener of this today's webinar, Dr. Junaid Tantray, for inviting me to interact with the participant and to give something about the animal model and the neuropsychiatric disorder research. So I'm thankful to all of you, and I also thankful to the participant who are giving their valuable time to learn something in the field of pharmacy. So thank you very much to everyone and welcome in this seminar and webinar. So today I'm going to discuss about the animal models and how these animal models can be useful for the neuropsychiatric disorder research and how we connect the preclinical research to the clinical application of that particular molecule. So briefly, I will tell you one story. Someday, some person wants to start research in his or her village. One old person told him that this particular plant have a activity, neuroprotective activity. So that activity or that utility of that plant should be explored by someone else, or it should be utilized for the human benefits. But that person didn't know how to showcase the property of that particular plant. How to test whether this particular plant have a neuroprotective property or not. So again, should I give this plant directly to the human being? Or is there any alternative? Since that particular plant ultimately going to be used by the humans. But that person didn't know even a single property of that plant except neuroprotective. That person was not sure whether that plant have a toxicity or not, how much quantity should be given, which route it should be given, which part of that plant should be given. So there are plenty of questions were there in his mind. Plant may be toxic, may not be effective. So he thought again, it is not wiseful to give directly to the human being. Then again there is a question, what should I do? But it was sure that it has a neuroprotective property because peoples were using from since antiquity. But in a modern era, the empirical knowledge is not acceptable until and unless we have a sufficient scientific data, scientific validity of that particular claim. So another person, he came to the one professor, and that professor told him that, why don't you develop a model? Or why don't you develop a neurodegeneration things in a particular non-human alternatives? It may be animals. It may be some cell line. That professor told him, okay, this is the particular model. You can develop the neurodegeneration using some chemical. And again, then you can test the activity of that plant. So ultimately what happened? That person developed a model using some chemical or other things. And later on, he studied the whole safety and toxicity, safety and efficacy of that plant in animals. So like that, here you can understand animal model, neuropsychiatric disorder, and how we can connect with them. Is my voice clear, sir?
Absolutely, sir. It's totally
Okay,
clear.
thank you.
PPT and voice also, sir.
Okay, thank you. So first we should know what is the animal model with special reference to the neuropsychiatric disorder research. So just now I told you a story. I think you got something behind the title of this webinar. So before going to study this safety and efficacy of any compound or any substance on human being, first we have to study or screen the toxicity and efficacy of that particular substance in animals. And that animals is known as actually animal model, including the procedure, how we are inducing that particular diseases, similar to the human being in each and every aspect, from starting from its causative agent up to the final damage of that particular parts or tissue of that human being. So we have to model the exact disease in a particular animal. So that is known as the animal model. Now, why there is a need? You can also understand with that story, because we have to, the first criteria of any medicine to be developed is it should have safety, safe. It should have devoid of toxicity. Efficacy comes later. First, it should have safe. So, ensuring the safety and efficacy, we have to study the molecules in animals. Once it is the safety and efficacy established in animals, then only we can go with the human trials. So how this connection between preclinical to the clinical is connected? The bridge is animal model. So today we are going to discuss about briefly, not too much, only briefly I am giving you how you can develop animal model, how you can study particular things using the different animal models. So in a Indian culture, there are so many things are there like that. Savidyaya abhimukte. This means it is the knowledge that enlighten us and education which liberates us from the materialistic world. So what is the education actually? What is the knowledge? Knowledge and education is different thing. So we are talking about the knowledge. Today we are gaining the education. But when we are applying that education for the benefit of the society, that is known as the knowledge. And until and unless you are free from any prejudice, you will not get or you will not justify with the knowledge which you have got. So with that quote, I would like to tell you briefly about the neuropsychiatric disorder. So neuropsychiatric disorder actually a broad term. It is not a small term. It includes so many things, both neurology as well as psychiatry. Means neurological disorder as well as psychiatric disorders. Both disorders included. And what are the things which are affected by these disorders in human being? So first is a behavior. For example, if you have seen a depressed person, so his or her mood is different from the normal people. If you have seen the epileptic person, so the behavior is different. And their emotion is also different. Suppose somebody have an anxiety, so that person can get irritated very easily. For example, somebody having an autism spectrum disorder or Alzheimer disorder, their cognitive dysfunction will decline. They have that they will not have that much of cognitive things. And why it is happening? Due to the dysfunction of the brain. It may be from the cellular level or from the molecular level. It may be due to the degeneration of a neuron or maybe the imbalance in the neurotransmitter level or other chemicals. So due to this, there may be a different type of the symptoms which may be cognitive. For example, memory loss, confusion, impaired judgment. Behavioral symptoms include agitation, impulsivity, social withdrawal. Sometimes you have seen people are very, very agitated. They are not in a society. They prefer to be in a isolated places. And sometimes people get irritated by small things. There is a swings in the mood. Sometimes they will feel very happy and immediately they will feel very sad. This is also known as the emotional problems associated with the neuropsychiatric disorder, such as such including depression and anxiety. And how these neuropsychiatric disorders are occurring. And let me tell you, nowadays the prevalence of the neuropsychiatric disorders has been drastically increased post COVID-19. Why? Because there are so many factors. There may be epigenetic factor, genetic factor, environmental factor, or some neurochemical factor. Let me tell you one thing. Nowadays, what we are eating, what we are watching, what we are listening, and what we are doing, all are adulterated actually. Our thoughts are also adulterated. Always we are thinking negatively. What we are eating, it has some chemicals which are damaging our neurons. Whatever we are watching nowadays, those pictures are not good. They are affecting our mental things, and what we are listening, what we are speaking, everything. In every aspect, we are having so many problems related to the brain. So before eating, before saying, before listening, before watching, we have to care. We have to mind the things. For example, we are not allowing any thief inside our home. Like that, we should not allow any bad thing inside our body. Maybe from eyes, from ears, from nose, from everybody, for surroundings. So we have to make the society like that, where harmony can be established, where good things can occur, so that your memory and your behavior, your mental status will be enhanced, or it will be maintained. So always you have to think about all these things. There are so many neuropsychiatric disorders: anxiety, depression, ADHD, Alzheimer disease, Parkinson disease, schizophrenia, autism spectrum disorder, so many. And all have three main components: behavioral changes, emotional changes, cognitive dysfunctions, and all these things. So why we are discussing about the neuropsychiatric disorder? Because the prevalence of neuropsychiatric disorder drastically enhanced. Currently, 970 million people worldwide affecting by the neuropsychiatric disorder. And from one in eight people are having the mental illness, mental problem. And due to this, the particular person will affect their daily life to life work will be affected, their families and whole healthcare system will have a burden. And ultimately, the country where the prevalence rate is high, they have to spend so much money on the treatment of these disorders. So ultimately, economy will be disturbed. So we have to take care of all these things. Ultimately, there are so many clinically available drugs are there, treatments are there. But still, the social stigma is the one of the problem. People are not easily going to discuss with the doctor that we have these problems. Medications are also available, but they have a side effects. Some behavioral therapies are also there. Some lifestyle things are also there. So moreover, we have to focus on lifestyle modification. Although it is not a lifestyle disorder, but so many factors are dependent on the lifestyle. For example, exercise. If you are doing exercise, it will release the endorphin. It will increase the serotonin level. Enrich environment, what we call it enrich environment. Diet. If you are taking a good diet, proper diet, balanced diet, fortified diet, it will enhance your brain power. Then social support. We are the social animal actually. So always we mingle with the people. We should not have to be isolated. So these are the different things. Nowadays, people are forgetting. There is a single families is there. They would like to be in a isolated places. So think about it. You are not going to do good thing for your health, your family, as well as your nation. You have to think about it. Now, why? Now we discuss about the prevalence rate and the brief about the neuropsychiatric disorder. Now, if we would like to develop a particular treatment or a proper diagnosis things, we have to understand the pathophysiology, pathogenesis of that particular neuropsychiatric disorder. Then only we can identify a particular target or particular key component which can be targeted by a molecule. So for that, how we can understand the normal pathogenesis as well as the treatment outcome of that particular compound? Through the animal model. So why there is a need of animal model? Since the animals we generally use for a particular disorder have a anatomy and physiology similar to the human being. Although some differences may be there, but most of the anatomy and physiology of human being should be similar to that particular animal model. So through the animal model, we can understand the basic science as well as we can develop the novel therapies for the treatment of that particular disease. We can also understand the normal pathophysiology, normal physiology as well as pathophysiology of that particular disease. And through the animal model, in the beginning I told you, we can also find out or we can also establish the safety and efficacy of that particular compound, which is not possible in a human being. And based on the animal model data, we can design a better clinical trial. As we know that there are different phases of the drug discovery and development. So before the clinical trials, there is a preclinical studies. Before the preclinical studies, we have to identify a target. We have to optimize the target. We have to validate the target. And we have to develop a molecule lead compound for that target. And from that, in vitro, in situ, in silico, then in vivo, these are the preclinical things. And after that, human trials. Phase 0 to phase 4. Like that, the drug is developed, discovered and developed. So animal model provide the pathway or direction for the better designing of the clinical trials. Now, what is the purpose of animal model in neuropsychiatric disorder? So already I told you, to understand the pathophysiology. Because we develop the disease similarly in animals, having a same or similar anatomy and physiology as compared to the human being. So it mimics the human conditions. We can explore the neurobiological mechanism involved in that particular diseases. And we can also study the developments of a embryo, or we can also study the development of a neurodevelopmental disorder, starting from the gestation up to the offspring. And how the neurodegeneration is occurring. What is the pathway? And what are the molecules involved in it? And how we can curtail that neurodegeneration progress. We can also understand drug development and testing by preclinical studies and behavioral analysis. We can also study the genetic component of that particular neuropsychiatric disorder by altering the genetic material of that particular animals. We can also study how the environment affects our genetic material. This is what called as epigenetic changes. For example, there is a well-known model for the autism spectrum disorder is maternal immune activation model. By giving the poly-IC in a pregnant woman at estrus day 12.5. So study shows that it induced the autism spectrum disorder in offspring in a F1 generation. Why? Because this poly-IC is a viral component which cause the inflammation or which cause the changes in the brain circuits of the developing embryo. So like that particular poly-IC or some other pesticides. Nowadays, farmers are using pesticide in a much quantity, which are affecting the pregnant women nearby of that particular farms. If a pregnant woman eating a vegetables or fruits having some pesticides or some chemical which cause this teratogenic effect. So the developing embryo will have a compromised brain function. Ultimately, later on, that particular child have a brain problem. So like that, how the environment is affecting the gene. So always think about the enriched environment. Enriched environment is what? Which supports you for the betterment of your life. For all aspect of the health, whether it's mental, social, spiritual, physical. Similarly, we can also discover or study the biomarker through these animal models. We can identify biomarker. I think you know what is the biomarker. Biomarker means which is the main component can give us the indication that if it is increase or decrease, the particular disease will occur. For example, if there is a decrease in the serotonin level, it means there is a depression. If there is an increase in a acetylcholine level, it means Parkinson disease is there. Now, we can also understand how we can translate the research. And we can also understand by the animal model the ethical consideration. Whenever we are performing any activity on the animals, we have to consider the 3R concept. Reduce, refine, replacement. So these are the purpose for the animal models. Now, question comes, what are the different type of the animal model? So based on your need, based on your circumstances, based on your laboratory conditions, and the availability of the particular component, you can develop a relevant model for your study. It may be genetic model by affecting the genetic makeup of a particular animals, because some neuropsychiatric disorder have a genetic component. For example, autism spectrum disorder, SANG3 gene. If this gene is not there, it is responsible for the autism spectrum disorder. Like then transgenic model, then CRISPR-Cas9 model. Precisely, we can alter the genetic makeup of a particular animals to induce a disease similar to the human being. Then pharmacological model. We can also induce the disease by giving the chemical or drug. For example, scopolamine. Scopolamine induce memory loss. Neurotoxins like MPTP. It causes the Parkinson disorder. Then lesion model, surgical and chemical lesion. So we can particularly affect the part of a brain. For example, olfactobulbectomy induce depression model. Like chemical lesion, so I already told you MPTP. Environmental model, like a stress, we can induce the stress. Chronic stress, restraint stress, unpredictable chronic mild stress, maternal separation stress, social isolation induced stress. These are the different things through which we can develop a model for a particular disorder. Here I am not talking about a specific diseases, because this is a broad topic. So I am giving you a brief about the animal models used for the neuropsychiatric disorder and how you can choose that particular model for your research. Then optogenetic and chemogenetic model. These are the newer things coming up. By using the light and chemicals, how we can affect the neurons. Because mainly neuron is responsible for the brain function and the chemicals involved, neurotransmitters. Most of the neurodegenerative disorders, neurons get damaged due to the different factors, including light, chemicals. Some are behavioral model, developmental model, and other model. For example, lipopolysaccharide-induced depressive model is there. Like I told you, MIA, maternal immune activation-induced autism spectrum disorder model is there. So there are like that, bacterial and virus can be used to develop a particular disease in animals, which is having the similarity to the human being. But next question comes in a mind. How we can validate that the model developed by us is have a same disease pattern, treatment pattern, behavioral pattern as compared to the human being. So for that, we have to consider three validity: phase validity, construct validity, predictive validity. Phase validity means if we have developed a model, that model should have a similar behavior as occurred in a human being. For example, epilepsy. In human being, epilepsy, there are 4 phases. Okay. Extension, flexion, tonic, cloning. So similar 4 phases should be observed in the animals. So how we can develop? By giving the PTZ or by giving the maximum electroshock. So same behavior pattern should be occurred in an animal so that animal have a good phase validity. Second is construct validity. Means what is the pathogenesis of that particular disease in human being? Same pathogenic pattern should be there in a human being. For example, in a depression, there is a decrease in the serotonin level. So in animal also there should be a decrease in the serotonin level. So this is known as the construct validity. If your model is also having a same pathogenetic mechanism, so we can say that model have a construct validity. It should not have some other alternative pathway to develop that particular disorder. Next is a predictive validity. Predictive validity means treatment response parallel human outcomes. For example, we always choose a one standard drug while making a group for a neuropsychiatric disorder or any research. Why? Because we compare our test substance with the standard drug. Suppose for the epilepsy, we are using phenotime, and phenotime decreasing the generalized epilepsy, all the 4 phases of the epilepsy. So our drug, if we are claiming that our test drug is having anti-epileptic property and we are using the PTZ animal model, so treatment should be similar to that drug or in parallel to that particular standard drug. For example, phenotime. So this is known as the predictive validity. So when we are developing any animal model, we have to consider all 3 validity: phase validity, construct validity, and predictive validity. Now, when we are using any animal for our research, so nowadays there is a laws in each and every countries and strict laws. We have to follow their guidelines. So we have to consider ethical things while performing any experiments on animals. What we have to do? We have to consider animal as human being. We should not consider them as an animal. We should not suffer them. We have to care. We have to provide the humane care to the animals without causing so much pain. And we have to also follow the guidelines given by the different guidelines of each and every countries. In India, CA. Earlier it was CPC SEA. Now it is CC SEA. Committee for the Control and Supervision of Experiment on Animals. It provides the guideline how to conduct the experiment, how to house the particular animals in an animal house, and what are the different other aspects, including transportation, number of animal to be used. So many things are there. Similarly, in USA, IACUC is also there. It provides the guidelines how to conduct the experiment by following all the guidelines. So we have to understand these guidelines. We have to follow these guidelines while developing any model or while doing any research on animals. Earlier, I told you 3R principles. Nowadays, people are not easily providing the approval for the research on animals. They suggest that you follow the 3R principle. Replace means, if possible, use the in vitro technique in place of the animal. Don't unnecessarily suffer or harm the animal. Second, reduce. Earlier, people were using number of animals in disproportionate. But nowadays, the committee is very strict. Whenever we are taking 8 or 10 animals, they are telling, oh, you reduce. As per the statistical significance, you have to take only 5 to 6 animals. Next is refine. Your procedure should be so much refined that the animal should have a minimal R. So these are the different ethical considerations we have to consider while doing experiments on animals. Then I told you earlier, animal model is not just the animals, or is neither just animal nor the procedure. It is the combination of both animal as well as the procedure to induce the disease in an animal. That is known as the animal model. So commonly in a neuropsychiatric disorder, we use rodents, rat and mice. Why? Because these are easy to handle. They have a similar anatomy physiology to the human being, and we can also manipulate the genetic things and environmental component in the mice and rats. Means we can also study the epigenetic studies. Apart from rodent, we can also use non-human primates like monkey for the complex behavior. Means the behavior which is not easily observable in the mice. Then nowadays, zebrafish is also commonly used by the neuroscientist. Some people are also using Drosophila and C. elegans for the neuropsychiatric disorder. But in a C. elegans, there is a difficulty in understanding the behavioral things. But Drosophila and zebrafish providing the good behavioral things as when we compare with the mice and rats. So these are the different animals which commonly used for the neuropsychiatric disorder research. Now, earlier we discussed about the different neuropsychiatric disorder. Now, what are the different tests we perform for the screening of a particular neuropsychiatric disorder? So these are the different behavioral tests for a different diseases. For example, anxiety and depression. So we generally use elevated plasmage, open field test, whole board test, light and dark test, force swim test, tail suspension test, sucrose piperine test for the depression, and other maternal separation induced depression, sleep deprivation induced depression, lipopolysaccharide induced depression. So there are different type of the model, but tests are only these. For the cognitive function, we can use the Morris-Water maze test to study the spatial memory. Then novel object recognition test to understand the cognition of that particular animal. Next is social behavior. We can also study the social behavior in the animals. using the 3-chamber apparatus test, locomotor activity by using the ectophotometer. So this is the brief things I discuss about you. And now the main focus of neuropsychiatric disorder is to study the animal behavior. Means how we can observe the animal behavior. So briefly we'll discuss about the historical part of the animal research, behavioral research. So actually these how animal are behave, we came to know from the Paleolithic era, where when the prehistoric hunter human ancestor understood that animal have a similar behavior to the human being. If there is a difference in the behavior, how we can understand? Darwin theory also told that how the modern animal behavior came in the existence. Ernst Mayr also described the typological thinking, or a concept about how human thinks and how it is different from animal thinking based on behavioral outcome. Ethology is the science of animal behavior study. Now, white man also developed a ethogram through which we can understand the behavioral pattern, especially in the pigeon. So like that, in a historical part, so many persons have also studied the behavior of the animal, and they have also compared the behavior of the animal with the human being, which was the basis to perform the behavioral analysis or behavioral research on the animals. The purpose of behavioral research simply the similarity, understand the similarity and differences between human and animal behavior. Prediction of unforeseen events such as earthquake. So for example, dog is very sensitive toward the earthquake or toward the smell. So before coming to the earthquake, dog can tell you that there is a earthquake is going to happen. So we can understand by the behavior of different animals. Diagnosis of disease, pathogenesis of disease, safety study of new chemical entities, and efficacy study of the new chemical entities. So through behavioral study, we can also do different things. Already I told you about different neuropsychiatric disorders. Now the question comes, whenever you are performing the behavioral study in the animals, so you have to take care so many things. It is not easy as you are thinking. Performing a behavioral study in an animal, it is very much tricky, as well as it needs so much attention and so much care. So when you are doing any animal research, so before the animal study, during the animal study, and after the animal study, you have to mind the pacer MI. You have to keep all these things in your mind. For what purpose you are going to perform the behavioral study? Which type of animal you are going to use? What is the environmental condition of your lab? It should be noise free. The animal should have a all phase predictive and construct validity. For example, if we are going to do the depression study, and we are going to perform the Morris Watermaid test or a tail suspension test. If we are providing or if there is so much noise, what will happen? Animal will get distracted. Sometimes if we are studying, we are having some task to be performed, but somebody knocked on door, what will happen? We will get distracted. So that will produce the biased result, false result. So the condition should be a soundproof room is required. Then experimental condition. The condition result generated by the lab which is situated in the USA and the result generated in the lab situated in India. The experimental condition may be different. So sometimes result may not come. So you have to optimize the things. you cannot exactly mimic the similar condition in the USA or in the India. You have to do some modification. So you have to consider the experimental condition as well. Rules and regulation. Whenever you are going to perform the animal research, you have to keep in mind the rules and regulation. Then, man, investigator and animal caretaker. Sometimes what happen when we are doing the research, if the person's behavior is not good, he is or she is irritated, he will put the cage like this. What will happen? Animal will have a different behavior. So this is also very much important. Then instruments, whatever instrument you have, or whatever you can modify or whatever you can do using existing instruments. So you have to plan the research like that. There are so many animal models for a particular one disease, but it depend on the condition, depend on the availability of the different instruments and different substances which is required to perform that study. So you have to consider all these things. Now we will discuss one by one briefly. So first is the elevated plus match. It is actually used for the screening the anti-anxiety agent and to study the locomotion. So here you can see that the principle. What is the principle? Principle is that height creates fear. So elevated plus match. Okay. Elevated plus match means what? I think the picture is distorted somewhat. So when we are in a height, we are on a height, what will happen if we see down? So we feel anxious. So like that, similarly, elevated plus match is developed to screen the anti-anxiety activity and to the locomotor activity. Similarly, there is a one apparatus which is known as the open field, and test performed using this apparatus is known as the open field test. It is also used to screen the anxiety, anti-anxiety agent and locomotion. So anxiety decrease the locomotion. Principle is that if the person having anxiety, so that person will spend much time in a periphery zone that is known as the thigmotaxic zone. If a person is not anxious, so the means the animal is not anxious, so the most of the time will be spent by the animal in a central position. Okay, so these are the different parameters. Then force swim test. Force swim test means this test is used for the actually for the screening of the depression. Okay, so in force swim test, what we do, we fill the water at a certain height and we put the animal on a that beaker and we forced animal to swim. So the principle is that depression decrease the mobility time. If some pupil is depressed or some person is depressed, so the locomotor activity or the mobility of that particular person will decline. So what we have to study here, if the person is immobile, means there is a depression. So in a force swim test, we keep the animal for 6 minutes and last 4 minutes will be counted for the immobility time. How much time that particular animal was immobile. If as compared to normal control, that animal has a more immobility time, means that animals have a depression. And if our drug is decreasing the immobility time and enhancing the mobility time, means that particular drug have a antidepressant activity. Similarly, we also use the tail suspension test to screen the antidepressant drug. Similarly, we tie the animal stiff with the help of tape, and at a particular height we hang the animal and we check the immobility time within a 4 minutes of time. Then social interaction test, which is commonly known as the 3-chamber apparatus and the test is a SIT, social interaction test. And this test actually used for the study of the autism spectrum disorder. So here there are 3 chambers. Okay, you can see there are jar, animal will be kept in the middle, and the principle is that cognitive impairments decrease the sociability. So in a autism spectrum disorder, the animal will be isolated. He will not, that animal will not interact with the novel mouse. We keep here initially familiar mice, later on novel mice, replaced with the novel mice. And we count or check the duration of interaction with the novel object or novel mouse. If the animal have autism, it will not interact with the novel mice, or the interaction time will be less. If similarly our drug have a effect, so it will enhance the interaction time with the novel mouse. So in autism, what happen? If we are meeting to the new person, so we isolate actually. Okay. If a person having autism, so the person will have a isolation. The person will not easily mingle with the new person. So same condition we generate here in animals. Next is a Morris-Watermaid test. Morris-Watermaid test actually used to study the memory function. There is a tank. The water is filled at a certain height. There is a one platform is there, which is invisible. And there we can divide the Morris-Watermaid, this water tank into 4 quadrants: north, east, south, west, and we left the animal in a different corner or different these directions, and we count the time to reach the to find out the hidden platform by the animal. That is known as the latency time. It's time taken to reach the platform. So principle is that memory dysfunction increases the distance traveled. If a person lost his or her memory, so he or she will not find a particular place uh, or in a certain period of time. It takes long time to reach that particular place. Similarly, we modeled and that Alzheimer disease in a this way. So we can study the memory using the Morris-Watermaid test. How much time the animal is taking to reach the hidden platform. So as compared to the normal control, Alzheimer mice having the Alzheimer disease will take much time to reach the hidden platform. But if our test drug having a ameliorative effect, so the latency time will be decreased. So like that we can study the Alzheimer like Alzheimer pathogenesis as well as drugs used for the treatment of Alzheimer disease using the Morris-Watermaid. So briefly, I told you about different behavioral analysis, their basic principle, parameter to be studied, and how you can model the different neurological neuropsychiatric disorder in animals. So based on that, we have performed behavior related things in our lab. Starting from my PhD up to now, we have performed neuropsychiatric disorders related behavior, mainly the elevated plasmage, open field test, FST, TST, SPT, sucrose preference test, Morris-Watermaid, novel object recognition test, SIT, social interaction test. And for that, we have got funding from different funding bodies, DBT, UGC, International Society for Neurochemistry, Switzerland, and IBRO. So we have performed the behavioral studies using the plant-derived natural component. Since we are in a Amarkantak area where there are many medicinal plants available, which are used by the tribal people of this area. So we are thinking that why should we should not go for the scientific validation of these plants and their active constant. So we are working on that. So in my PhD, actually I worked on monocule. and the model was LPS-induced anxiety and depressive-like behavior in mice. So we design experiments where up to 2 days we have given the pre-treatment of honocuol, 3 days, and at 0 hours, on third day we have given the LPS. LPS means lipopolysaccharide, which is a bacterial endotoxins. Okay, so it causes actually the research is claimed that LPS causes the sickness behavior, anxiety, and depression in a human being. Okay, so we have given this injection, and at 3 hours of this injection, we have tested for the elevated plasmage. After 4 hour OFT, and after 24-hour FST, after 28 hours TST, and after this at last, we have performed the sucrose footprint test from the 24-hour to 48 hours. And this is the validated model. So here we have taken the different time zone, different time intervals. Why? Because one test should not interfere with the have an interference with the other test. So we have kept the different time intervals for performing different tests. So we actually perform the elevated plasmage activity, and we have considered three parameters: number of entries in open arm, closed arm, and time spent in open arm. So in a plasmage, there are two open arms and there are two closed arms. Okay, so if person having anxiety, it will spend much time in a closed arm, means he or she needs support. So similarly, when we kept the animal on open area, on a central area, so animal having anxiety will always uh, spend more time in a closed arm. So here you can also see that these LPS mice, okay, these LPS mice have spent less time in an open arm, okay, and spend more time. Number of entries was also less. Time also time and entry in open arm was less. So most of the time spent by the anxious mice in a closed arm. So we can compare that. And similarly, the high dose of this LPS provided the protection against the LPS induced anxiety like behavior when tested in the elevated plasmids. So we have performed these things. Similarly, we also perform the anxiety-like behavior of monocuol in OFT, open field test. So in open field test, okay, this open field test, we divided that open field into 16 quadrant. Okay, earlier I told you 16 quadrant. And the outer quadrant were considered as the peripheral zone. And the middle part, 4 zone, were considered as the central zone. So we have uh, counted the frequency, how much times the animal crossed the central zone and peripheral zone. So here you can see that this LPS treated mice showed the decreased level of the central crossing and a peripheral crossing and rearing. But later on we find that there is a high dose of the honocuol showed the protective effect against the LPS induced decrease or anxiety-like behavior. Similarly, we also perform the depressive-like behavior of that same drug, honocuol, in a LPS-induced depressive-like model. And here we found that when we are giving the LPS, so it increases the immobility time. Means the animal showed the depressive-like behavior when we tested in the FST. So immobility time will increase as compared to this normal control group. But whenever we have observed the behavior in a pre-treated animal with the honocuol at both the doses, so we found a significant decrease in the immobility time. So it shows that honocuol has some protective effect against the LPS-induced depressive-like behavior. Similarly, we have also performed the tail suspension test using the same drug are in a same animal after 28 hours of the LPS injection. So we found that there is also a significant increase in the immobility time when tested in the tail suspension test. And we also found that both the doses of the honocure decreases the immobility time. Thus, we can say that this honocure at both the doses showed the anti-depressant-like effect by reducing the immobility time in a LPS-induced depressive-like model. And these results actually are supporting the earlier studies, which says that honocure also have a protective effect against the uh, means anxiety and depression. Not the honocure, it's a decoction. It is a plant, means it is the uh, component obtained from the Magna officinalis, which is obtained in a occur in a China actually. So they use decoction to treat the depression. So later on, we performed the study and found the protective effect. We also checked whether the honocule have an anhedonic suppression effect on SPT or not. Because if person having a depression, so it has an anhedonia. Anhedonia means loss of pleasure in a normal activity. So person having a depression, it has a less pleasure, okay, loss of pleasure to perform the normal activities. So what we have to do, we have to test whether our component have a antidepressant effect or not. So what we have to do, we have to perform the sucrose preference. Sucrose preference means we have given first the 1% sucrose to the animal. So animal acclimatize that we have to drink this sucrose. Okay. Later on, we replace the bottle in kgs. At one side sucrose and one side normal water. And we measure the water. And after 24 hours of the duration, again we measure the volume consumed by the animal, normal water as well as the 1% sucrose solution. And then we calculate it and we find out the percentage sucrose preference. So if the person having a depression, so it will consume less sucrose solution. So similarly, we have found in a LPS animal, there is a decrease, significant decrease in the percentage sucrose preference as compared to the normal control. So here we can say that LPS induces the depression. Later on, we found that both the doses of the honocure significantly increase the percentage sucrose preference. So here we can say that this honocure also provide the protective effect against LPS-induced depressive-like behavior by increasing the sucrose preference. Then later on, we also perform a study related to the different animal model. This animal model was the sleep deprivation-induced depression as well as cognitive dysfunction. So here we found that escalating improved memory function in sleep-deprived mice by reducing escape latency in Morris-Bottomley test. So we have performed the Morris-Bottomley test in an animal subjected to the sleep deprivation. So in our experimental design, we have adopted the modified platform method, where we kept the animal on a platform, 3x3 size platform, and we put the light 24 hours, and like that we deprived the animal by sleeping. That is known as the sleep deprivation induced cognitive dysfunction. And we have given the pre-treatment of 14 days pre-treatment of the escalating, and up to 14 days, and after 14 days 5 days sleep deprivation. So after total 14 plus 5 19 days, After 19 days, we have performed the different behavioral studies, including the Morris-Bottom-Age test, to check what is the effect of escalating on sleep deprivation-induced cognitive dysfunction. So we found that the sleep-deprived animal have a significant escape latency. Means it take the much time to reach the hidden platform. Because means the cognition of the sleep deprived animal were compromised. So later on, we have given the pre-treatment of the escalating and subjected to the sleep deprivation. So we found that this escalating significantly decreased the escape latency. Means it took less time as compared to the sleep deprived animal to find out the hidden platform. So like that, we can say that escalating has a the memory enhancing property. Recently, we have completed one project funded by the International Society for Neurochemistry, where we have studied the effect of mangiferin and escalating on poly-IC-induced depressive-like behavior and cognitive dysfunction, as well as autism spectrum disorder. So for autism spectrum disorder, we have developed a model, poly-IC-induced maternal immune activation. Means for that, we have given the poly-IC at 20 mg per kg body weight through IP route to the pregnant woman. So first we have kept male and female mice for the mating. Once the mating is confirmed by the checking the vaginal plug, and after confirmation of the pregnancy is done, that day is considered as a 0 day. So after 12.5 hours, exactly stress day 12, we have given the poly-IC to the pregnant mice through the IP route. So, and after that, we kept the pregnant mice as it is and allow them to deliver the pups. Once the pups were delivered, we waited for the 1 month, and after 1 month, we performed the different behavioral studies, including the anxiety-related, depressive-related, and at the 7 to 8 weeks of the age of that offspring, we perform the social interaction test, which is used especially for the autism spectrum disorder screening. So in that, we tested the time spent by the animal with the novel mouse, test mouse and novel mouse. So we found that as compared to vehicle, this poly-IC treated pregnant mice offspring showed decreased social interaction with the novel mouse as compared to the normal pregnant woman offspring. Later on, we have found that when the pre-treatment and same times actually, when the pregnancy is confirmed and up to 12 days, we have also given the pre-treatment of mangiferin and esculating to the pregnant woman through the oral route. Okay. So basically we divide it into 4 groups. First is a pregnant woman receiving vehicle. Second group, pregnant woman receiving the poly-IC. Third group, escalating low dose and challenge with the poly-IC. Escalating high dose challenge with the poly-IC. So like that we have grouped the animal and waited for the delivery of the pups. And after the 7 to 8 weeks of their age of the offspring, we have performed the social interaction test. So we found that the pregnant mice, which were pre-treated with the escalating low dose and high dose, showed the significant increase in the time spent with the novel mice. So here the conclusion is that escalating improved the social interaction in offspring of poly-IC injected female mice by increasing time spent with the novel mice in SIT, social interaction test. So like that, we perform different behavioral studies, starting from the anxiety-like behavior, depressive-like behavior, locomotor activity, then this memory test, water maze test, social interaction test for the autism spectrum disorder. So like that, we have done so many behavioral studies, and we found that there are so many challenges are also there while performing any behavioral studies. It is not the clear-cut indication that a particular drug provide the protective effect. There are so many challenges while we are translating the data to the human from the preclinical to the clinical. What are the reasons, or what may be the prospective or retrospective reasons? The reasons are species differences. So many researchers have also indicated this thing. For example, if you are doing the behavioral study, so you can you have to choose the or you should choose the C57 mice for the behavioral study as compared to the Swiss albino mice or the other mice. Why? Because it is sensitive. C57 black mice is very sensitive toward the behavioral result. We also perform these activities on the C57 BLG6 mice. Why? Because the species, same strand, different species, have a different anatomical and physiological variations. So we have to consider these things while selecting any. The next important thing about the neuropsychiatric disorder is there is a complex pathogenesis. I mean, so many things are involved. For example, inflammation, oxidative stress, nitrous stress, neurochemical things. So many things are involved. There is a complexity of the neuropsychiatric disorder, which affect the data or behavioral analysis. Then, if we would like to really translate the data from the animal study to the human study, so we have to include the different modern techniques. Means we have to develop a such model which are multi-dimensional. Okay, so that each and every aspect can be covered in that. Okay. Now we can advance the animal model by using the CRISPR-Cas9 techniques, where a precise genetic alteration can be made. Okay, because in a normal knockout and knock-in model, the whole gene will be removed, or whole gene will be inserted. But in a CRISPR-Cas9, a specific the part of the gene we can alter. Okay, so like that specificity will be there. Okay, now optogenetic and chemogenetic techniques we can also include in our animal model, so that the neural activity can be controlled by using the light and the chemical at a specific site, actually at a specific part of the brain. And same time we can also perform the, or we can also connect in vivo imaging. Real-time monitoring of the brain function can be done with the animal model. So these are the advanced techniques available nowadays. Earlier, these were not there, but nowadays you can utilize these techniques. So this is all about the animal research up to now. What things left that can be included in a animal model while doing your research? So you have to think all the aspect of the disease. For example, genetic, epigenetic, environmental, pharmacological factor. Since the neuropsychiatric disorders are having a complex pathogenesis, so when we are developing any model, we have to enhance the model relevancy by combining genetic and all these factors. We can also collaborate with the other persons. Suppose we are doing on chemical induced neuropsychiatric disorder model. Somebody are having expertise in knockout genetic modification animal model. Somebody is having a optogenetic like facility, so we can collaborate with them, and a concrete result can come by doing the research, or by doing the same research using the different models. Okay. And nowadays we can also integrate the new techniques, innovative techniques like optogenetics, chemogenetics, and real-time imaging of the brain function. So like that, using AI also we can use to develop the animal model. So like that, we have to think in a future while developing any animal model for neuropsychiatric disorder, because every model have some limitation, some advantage as well as disadvantage. So which animal model is suitable for us? We have to think about it. So today's talks takeaways are there. Animal behavioral research is an integral part of neuropsychiatric and other brain disorder. To understand the pathogenesis, to ensure the safety and efficacy and correlation of human disease with physiological changes. Animals should be considered as humans with proper rules and regulations for the benefit of society. Animal models provide proper direction for human research and its outcome for better clinical application. So we cannot go without the animal research, especially in case of the neuropsychiatric disorder, because we cannot measure human behavior by some other alternative. We have to depend on the animals. So this is the main things. So while performing the experiments or while developing the animal model, we have to take care all aspects of the behavior as similar to the human being. So these are the things which we discuss about today, that how animal model can be useful for the neuropsychiatric disorder. So I always quote these things in my talk or everywhere, that Swami Vivekananda ji always told about that whatever goal you have decided to attend that goal, you have to do उत्तिष्ठ जागृत प्राप्त वरानी बोधा, उठो जागो और ध्येय की प्राप्ति तक मत रुको. Arise, awake and stop not until the goal is reached. So whatever today you have achieved, it is not your ultimate goal or ultimate ध्येय laksh. You have to go beyond that. Once things you have completed, just set a different next goal and achieve that. So then only you can achieve your life. So ultimate goal of life is to be happy and to make others happy. This is the harmony of today's world actually, and an ancient world as well. So we have to learn from the ancient things to make the modern world happier, to make a harmony. So we have to make a harmony with the animals as well. We have to think like a human being. So thank you very much from my side, and whatever research I have presented that were funded from the UGC, International Society for Neurochemistry, and we also thankful to the our home institution, Indira Gandhi National Tribal University, for supporting me in all aspect to improve myself day by day. And this is the NPRL team. These are three my PhD students, Ramu Singh, Angelina, and Madhvi. Two have submitted, means they have given their pre-PhD seminar. Now soon they are going to submit their thesis. And my area is neuropsychiatric disorder. So we are exploring the different plants and their active constant for ameliorating the neuropsychiatric disorder. So thank you very much for giving me this opportunity, and thank you very much for listening me patiently. If you have any question, you can ask me, and you can also reach me out using a different social media, and there's a scientific media profile. So thank you very much. Thank you.
Thank you so much, sir. It was nothing sort of exceptional. आपने बात की, sir. You have talked about how prevalence will affect the economy of country. You talk about animal models. You talk about importance of animal models in research. You also talk about purpose of animal models, types of animal models, criteria, and most important, sir, you talked about what we eat and what we watch is having impact on our mind and brains, which is very important thing. One of the getaways, sir, and we have received numerous questions, sir. But out of that, we will, as we have a time limitations, but I have received some few questions. One of the question from name Prime Mini, sir, how we can evaluate the locomotion by using plus maze? Please answer. ऐसा लिखा है, sir.
Okay. So actually, from the elevated plus maze, we cannot actually exactly not locomotion activity. We can observe the time spent in a, especially in a open field model and in a means this closed arm and open arms. But for more specifically, we use the open field test as well as ectophotometer for the locomotion.
I hope that person has heard this answer. And sir, next question we have received from Naveen Tripathi. Sir, my question is that what are the various criteria for choosing the rat in place of mice in depression models, or how we can decide to choose rat or mice in our depression models?
Okay. First thing is that availability of the animals. First thing. Second thing, how much tissue we would like to take from after the sacrificing the animal. Okay. Suppose you would like to take you need means much tissues for the biochemical as well as molecular study. So we supposed to go with the rat model. Okay, so there is a no difference between rat and mice. It is just the size matters. Okay, and it is your expertise. Some people are not familiar or not easy or have a comfort zone with handling the rat, so they prefer to the mice, nothing else. So you can choose any of them. And second thing, third thing is that uh, the size of the apparatus for the mice as and rat are different. So for mice you need bigger size of the elevated plus maze open field test. So if you can manage, you have enough space, then you can go with rat. Otherwise, you can opt for the mice. So there is a no difference such as.
Right, sir. So one of the question from Swati Prakash. Sir, can you please tell about chronic stress-induced depression model? How many minimum parameters will validate the study protocol?
Chronic induce means there are different ways to induce the chronic stress. Okay, so you can also use the restraint stress, or the most widely used stress induced depression model is UCMS, unpredictable chronic mild stress. So you can choose a different, but as per protocol minimum, you have to take 8 to 10, matlab 1 week, at least 1 week model. So in 1 week model, you can choose different unpredictable chronic mild stress, which comes in a human life actually, because we are mimicking the depression in an animal. So it is not compulsion that you have to use all the chronic stressor. You can choose either of them which are suitable for your lab. So you can better to go with the unpredictable chronic mild stress. So some takes 14 days, some takes 8 days, some takes 5 days. So it depends on the how much duration you are giving the stress on in a 1 day. So likewise the time will reduce or increase. If you are giving less stress at 1 day, so time will is increase. If you are giving more stress on 1 day, so time will decrease. Okay, like that you can choose. There is a no problem.
So this is after a long time. We have received so many questions. Just two, three. We are picking last of them, and one is Gauri from Gauri Gawanda. Sir, kindly tell us about the chemical parameters to check in anxiety and depression activity.
Chemical parameter. chemical parameter actually in a anxiety, there is a decrease, there is a change in the adrenaline level actually. In anxiety, there is a more adrenaline level. So you can check adrenaline first. Second, nowadays there are ample of research shows that there is an inflammatory pathway in a commonly inflammatory pathway, oxidative stress. So you can go with the pro-inflammatory cytokines, interleukin 1 beta, interleukin 6, TNF alpha, interferon gamma, as well as anti-inflammatory cytokines like IL-10. Okay, so you can go with that. Nowadays, it is also mentioned that BDNF level is also affected, which is responsible for the synaptic plasticity. So you can also measure that. You can also measure the serotonin level because this buspirone is a 5-HT 1 alpha agonist. Okay, so you can also measure the 5-HT level using the and their metabolite using the HPLC ECD detector. So you can also measure in the blood as well as the brain, amygdala. Okay, like that.
Right, sir.
Amygdala is the main things for the response for the anxiety. Okay.
Anxiety. Sir, one more. This is from Shahid Nazir Wani. It was quite insightful session, sir. Please give some insights on what are different animal models for drug addiction and withdrawals, especially opioids, and what are the advancement in this area.
Actually, I don't have expertise in a addiction
Drug
related
addiction.
model, but I'm thinking to work on alcohol induced model. So one thing I would like to discuss what our B.Pharm student recently has done. They have used the alcohol, okay, and in a geographies means they have from 0.1%, 5%, 1%, 1.25%, 1.5%. Like that, they have increased the concentration of
Dose.
the alcohol in a zebrafish and up to I think 12 days exactly I forget to up to 12 days they have escalated the dose of the alcohol and they tested in a zebrafish and they have found the good result by increasing the dose of the alcohol. They found there is a cognitive decline in the alcohol induced model in a zebrafish. If you need, you can write to me. We can share you the details of that protocol. And for a withdrawal we have not worked. What I think ketamine and opioid induced withdrawal models are there, but exactly I don't know about them. If you want really, I can read and I can we can discuss.
Sir, last question is there from Mayank Sharma. Sir, how we can determine the biomarkers which could be key players in neuropsychiatric disorder?
See, before analyzing or before measuring the biomarker, you have to select the disease. See, there are different disease. There are different biomarkers. So you have to find out the specific biomarker for a specific disorder. For that, you have to understand the pathogenesis, exact pathogenesis. The problem in the neuropsychiatric disorder is that there is a complexity. We cannot say that this is the particular biomarker which is responsible for the particular neuropsychiatric disorder, except some few disorder like depression, anxiety, Alzheimer disease. So, for example, in a depression, there is a decrease in the 5-HT level in both blood as well as the hippocampus. So, you have to take out the hippocampus as well as the blood, and in blood you can measure the 5-HT level. Similarly, in case of the Alzheimer disease, you can also measure the tau level, A beta protein level. So like that, you can measure biomarker in a particular tissue or in a particular fluid. Okay, like that you can do. आप
Uh,
हाँ.
Thank you so much, sir. We, as you can see, sir, we have n number of questions. It means that it has gone very well to the audience. Or thank you once again. As we conclude 60th SPSR webinar, animal models and neuropsychiatric disorder research, building the bridge. We have delved into the critical role that animal models play in advancing an understanding of neuropsychiatric disorders. Our discussions have highlighted the ongoing challenge and the innovative approach being employed to refine these models, ensuring they more accurately reflect human activities. Let's carry forward the knowledge and inspiration garnered today as we embark on a journey to continue push the boundaries of research. We are truly thankful to the contribution of our keynote speaker, uh, Dr. Kunjbihari Sulakhiya sir, whose expertise provided us the valuable understanding of animal models and neuropsychiatric disorders, building the bridge. We acknowledge the convener, Mrs. Monika Sabharwal ma'am, for a critical role in organizing the event and our knowledge encouraged cooperative learning. We are grateful for all significant involvement in the 60th SPSR webinar. Your engagement, participation, and input have been essential, and we eagerly anticipate collaborating on upcoming initiatives aiming at enhancing healthcare and well-being for everyone. Thank you once again to the each and every participant for being there patiently and contributing more improvement in the profession. Join us to the upcoming 61st SPSR webinar, Advancement in Dosage Forms for Elderly Patients. Our esteemed speaker for this event will be Dr. Liu Cal Bin, Head of Department Pharmaceutical Technology and Industry, University of Saigeriya, Malaysia. To register, please subscribe the official YouTube channel of SPSR. Once again, thank you so much, sir. It was also a brainstorming session for me. I will definitely, sir, contact for further more details. And thank you once again, sir. Thank you so much.
Thank you, and thank you everyone. If anybody have a question, you can share my email ID, no problem. Okay, they can ask me later on. I don't have any problem. Okay. You can
Sure, sir.
also note down naiparkunj@gmail.com. naiparkunj@gmail.com. So if you have any question related to the thing, you can ask me. So thank you very much, Junaid ji and Monika madam,
Pleasure, sir.
Alok sir, for giving me this opportunity, and thank you very much to all the audience for listening me carefully. If you have any question, and if any I have whatever I can do, I did my part. Okay, so thank you very much. Have a nice day.
Thank you for being gracious.